Good afternoon, welcome back to Virology. Today we are starting the second half of the course where we're going to talk about viruses infecting host organisms. And today we're going to talk about principles that we need to understand what I'm going to talk about for the rest of this course. In a host in nature, the virus genome needs to endure. It needs to get into the host, it needs to multiply, it needs to get out. I say the genome because the shell is just a carrier. It's all about the genome. If the genome doesn't persist, it ends. And that's not something that we needed to deal with when we talked about virus infection of cells in culture. As you can see by this dish here, all we did was add virus, it would reproduce, kill the cells, and that's the end of it. It doesn't go anywhere else. So obviously there's a big difference in what we've talked about so far and what we're going to talk about for the rest of this class. On the other hand, there are similarities, of course. What happens in cells in culture probably to some extent happens in the infected host. And in both cases, the virus needs to get into the cells, they need to get out. But for the host, there's an additional complication, and that is the virus has to get in the host, and then it gets in cells, gets out of those cells, and then gets to another host. So the very first day, I said we live and prosper in a cloud of viruses, and that is largely because most infections have no consequence to us. Many of them are what we call inapparent or asymptomatic. These are two very important terms. You should understand what they mean. And so let's define the terms of infection. A sign of infection is evidence of disease that can be observed by others. So if you have a rash, that can be observed by someone. If you measure fever, weight loss, CD4 lymphocyte counts, all of that can be measured by others so others can observe it. Those are signs of infection. And a symptom is what you feel, and that only you can sense. And if you have a stomach ache, unless you tell someone, they won't be able to know it, and they can't even measure it. So that is a big difference between symptoms and signs. Don't use them loosely. They're very precise. So when we talk about asymptomatic infections, that means you don't feel anything. It doesn't mean nothing is going on in you. Right? We could measure things happening. We could measure fever, for example. You may not feel it. And people have different levels of sensitivity to what they feel. For someone, a headache is horrible, and for the next person, they don't even feel it. It's just so interesting how it varies in a population. So asymptomatic infection is defined as one where the virus is in you reproducing and you don't feel anything. Whereas signs, of course, are what others measure. Inapparent is another way of saying asymptomatic. It's an inapparent infection to you, but in both cases, things are going on. Okay, so an example is West Nile virus. By the way, not present in the US before 1999. Came into the US, came here in New York City, probably brought in by a traveler, and then in the next 4 years spread across the whole US. It started in New York, and you can see by these maps of the US, the different colors are extent of virus in different states, and now it's throughout North America. By October 2014, about 1 million people were infected. We know this because we did serological surveys. A serological survey means you take blood from people, you make serum from it, and then you look for antibodies against the virus using methods that we've talked about before. So we know about 1 million people were infected. In a serological survey, by the way, you can never check everyone. It's impossible. We can never check 1 million people, unless there's a pandemic maybe. But what you do is you pick small numbers of people in different areas that have different representations for different demographics, and then you use statistics to extrapolate. So we say 1 million people were infected by October 2004, but only 20% had febrile illness. 20% of the people. And in about 1% of the people, they developed central nervous system disease, which is very obvious because you have either paralysis or cognitive issues and so forth. So that's quite clear. And typically if you have a fever, you feel it and you can measure it, and you may go to a physician and it gets recorded. So that's how we know that there are so many asymptomatic infections, right? 80% of the people in these 5 years or so did not have any symptoms because they didn't seek medical care and didn't think anything was wrong with them. So these are typical parameters for what we call acute virus infections. That is, virus infections that begin and end in some way. They don't go on forever, and they end in either you getting better or dying. But a persistent infection goes on for the life of the host, and we will talk about both of those in more detail later. But acute infections are a problem because they often have a lot of asymptomatic infections. Okay, so the other issue or the other aspect of an infection is the incubation period, highly misunderstood by many. The definition is the period before symptoms of a disease are obvious. Symptoms, what you feel, not what we can measure, all right? So you're chugging along and you get infected, you don't know that, but suddenly you're vomiting and you have diarrhea. Those are the symptoms. That's norovirus gastroenteritis. However, in the incubation period, as you can see from this picture, the virus is multiplying in you. You don't know it because you don't feel it yet. And the host is responding. We could measure within the first few days some cytokine responses, not antibodies and T cells yet, that come later, but yeah, we could see the host is responding. And here's the key. The virus may or may not be transmitted during the incubation period. This is very important. That's not the same for all viruses. For Ebola virus, it is not transmitted until you feel like hell. It's not transmitted during the incubation period. So another way you could look at the incubation period is the period until you feel like hell, right? But the period before symptoms appear. And for other viruses, they are transmitted during the incubation period. SARS-CoV-2 for sure is transmitted during the incubation period. And in addition, these asymptomatic infections that I just described, you may never develop symptoms even though you're infected. Many viruses do that, and about 20 to 40%, depending on the study, of SARS-CoV-2 infections are asymptomatic. The virus is multiplying, it's being transmitted, and you don't even know it. So that's a big problem for a virus, right? That is called community spread. SARS-1 did not do that. There was no asymptomatic transmission or pre-symptomatic transmission. When you got sick, that's when you transmitted, but you were so sick, you went into a hospital, and there we can limit transmission. And so that's why that never became a pandemic. That was easy to stop, right? So that's incubation period. And here are some periods of some viral infections. So incubation periods vary. They could be short, a day or two. Influenza's quite short. Rhinovirus, Ebola. Ebola goes out to 21. And all the way up to 50 to 150 days for warts caused by papillomaviruses. SARS-CoV-2 is 1 to 14 days. Incubation periods are difficult because you're depending on clinical observations. It's not easy. And if you ask people, when did you first feel sick? That's opening a can of worms because people can't remember anything generally. So you have a lot of variability there. The only way you can really pin down an incubation period is to do a challenge trial, which we don't do a lot for ethical reasons, but there's been one done for SARS-CoV-2. And if you infect 50 people, you get a range of incubation periods. Everybody is different. Everyone's different genetically, and it influences their responses to infections. So viruses with short incubation periods, typically what's going on there, the virus enters and reproduces close to where it enters, so it doesn't have to go far to produce the signs and symptoms of disease. Let's say symptoms, because this is incubation period. And then viruses with longer incubation periods, they may have to travel somewhere before they produce the symptoms of infection. And then one more term here that you'll see, I may not use it again, or maybe one more time, is prodrome. That's the symptoms before the ones that are characteristic of the disease. So many viruses will give you what we call a flu-like illness, fever, muscle ache, malaise. Many viruses do that because those things are caused by interferon responses. So they're generic, let's say. And then there's a point where you get something that is very specific for the virus, like paralysis for poliovirus, or the rash of measles, say. All right, so the symptoms up till then is called the prodrome. Prodrome is a precursor to the symptoms characteristic of the disease. All right, so that's incubation period. A couple of other terms I want to tell you about. I actually added this slide during the pandemic because these were thrown about wildly by everyone who didn't know what they meant. And they assumed if they looked at it at Wikipedia, it was correct, but not always. Morbidity, mortality, incidence, and fatality. Okay, and we're going to give some examples of fatality. So these are icons where each person is 10,000 people, and the two colors, orange and red, the orange ones are people who are infected as defined by being PCR positive, and then the red one is people who have signs and symptoms. So signs is, PCR is a sign, and a symptom would be they're reporting that they're ill. So incidence is the number of people infected over the number in the population with a time component. So in that example on the left, it's 25%. Any particular part in time, right? You say, okay, in this month, the incidence was. It's not a whole year. Well, it could be a whole year if you define it as a year. And the morbidity rate is the number of people ill over the number in the population. And that's the second one. It's 10% in that example. So you have people who are the red ones because they have signs and symptoms. Those are the ones who are ill. Mortality rate, this is the one that the press bandied about extensively to scare everybody about the pandemic, is the number of deaths over the number in the population. And the case fatality rate, another one that was thrown about, the number of deaths over the number of confirmed infected. And these are difficult numbers to get. You never get all of them. You never get all the deaths because not everyone who dies of COVID has on their birth certificate, right? On their death certificate, COVID as a sign of death, as a cause of death. So very hard to confirm. You may have COVID and die of a heart attack, so they'll put heart attack as a cause of death. What you should do is put heart attack due to COVID or something like that. And then we have the infection fatality rate, which is the number of deaths over the number of actual infections. So what's the difference between CFR and IFR? CFR, you look at all the PCR positives in a region and you say how many people died and you simply do the math. But the number of actual infections is a statistically extrapolated number because confirmed infected isn't everyone. You never get everyone. So IFR is thought to be closer to reality, but in fact it is a statistical extrapolation, which in biology is usually a problem. Always a problem in biology. So let me show you why the CFR doesn't tell you everything. These are early numbers from China, fatalities caused by SARS-CoV-2 infection. On the left, these were 44,672 cases, the first ones in China in 2020. And you can see 44,000 cases, 1,000 deaths, so the CFR is 2.3%, okay? That's not a biological constant, because if you look down the table, if you stratify it by age, you can see that very young kids, well, in this sampling, there weren't any deaths. Kids do die, though. Mark my word, kids do die. This was just early on and not many cases. But if you go down in age, it goes up to 14.8% if you're 80, with different numbers in between. So age is obviously a factor that contributes to mortality. It's not just the virus. Yet early in the pandemic, it was just the virus. That's all that mattered. On the bottom is sex. Men are two times more likely to die than females of COVID. And then on the right is a graph showing you, depending on where you were in China, the CFR varied. The blue line, and it's varying with time, January through February of 2020, very beginning of the pandemic. This is the deaths over the cases, right? CFR. Wuhan, huge, 20-some%, and then it goes down. And at the same time where Wuhan is high, other parts of China are low. Why is that? Hospitals got overwhelmed in Wuhan. They are full. People are dying on the streets. They didn't know how to take care of patients. They figured it out. Other parts of China, there weren't so many cases. The hospitals could take care of it. These are the factors you have to consider when you're talking about CFR. It's not just the virus. Oh my God, it's a virulent variant. It's got a CFR of 20. Well, give me the other variables as well. Make sure you look at the whole story. This is what we're going to see over and over again with this pandemic because it was talked about so extensively. Now, another topic that is widely misunderstood is what we call the basic reproductive number, R naught, which no one had ever heard before except in the movie Contagion. You guys have seen that movie? We're going to have an extra credit at the end of the course about that. We'll see how much you understood about the movie, okay? So the basic reproductive number is defined as the number of infections that one person can generate. So if you're infected, how many other people can you infect? And this needs to be calculated before there's any population immunity, before there's any interventions, if you want to get as close as possible to what the virus can do. And that's the formula. R naught for tau is the probability of infection given contact. C is the average duration of contact between the infected and the uninfected host. And D is the duration of infectivity, how long you are actually infectious. So there's a human behavioral component here, obviously. Probability of infection given contact, the average duration of contact, et cetera. So the reason this is a useful number is because if you can get it less than 1, the epidemic will stop. If you can tell people to stay at home or wear masks, you maybe could get it less than 1 and you can end an outbreak. But if it's greater than 1, then you're having an outbreak. If it's much greater, you're definitely having an outbreak. So, early on for SARS-CoV-2 in China, before any population immunity, before any interventions, it was calculated, I think, 2 to 4. So one person could infect 2 to 4 other people. Now, obviously, it's influenced by how much time people have contact with each other. So if you're in your family, it's more likely that you're going to transmit to other members of the family because you're together more and so forth. The length of the infectious period, the period that you're shedding enough virus to transmit to another person. And as I said, interventions like sheltering and masks can affect it as well. So it's not a constant for a pathogen. People think it is. And people have tables of R naughts for different viruses. They're bogus because it depends where you are and what's going on, the R naught, right? So it's not a constant. You can change the R naught for a pathogen. And so during this pandemic, people said, oh, this variant has an R naught of 10. It's going to kill everyone. And first of all, it's no longer the R naught once there's population immunity and interventions. It's now called RE. Did I have that on the previous slide? Yes, RET, R effective. So you have to account for that. But it is influenced by what you do and also by human behavior. I remember I'll get you just a minute, in the UK when the Alpha variant first came out. Oh my gosh, this virus is spreading like crazy. It's got an R naught twice as much as the original virus. They were just coming out of lockdown. Everybody was going to bars and theaters. Don't you think that's going to stimulate spreading? And none of the R naught calculations took that into effect. So be really careful about that. Let me move through this and then I'll get to your question. So R naught, the other problem with R naught, it's calculated from models. And everyone that does their R naught uses a different model, so you can't compare them. It's like apples and oranges. So, and by itself, it doesn't tell you how fast an infection will spread. It just tells you how many people one person can infect. So for SARS-2, it's 2 to 3. One infected person can infect 2 to 3 others. I don't believe any subsequent R naught calculated during this outbreak for SARS-CoV-2. I think most of them did not consider human behavior in really understanding why one variant spread faster than any others. Okay, so beware of R naught. And this idea that a virus is mutating, which came from contagion, right? The virus is mutating and the R naught is increasing. You have to be really careful to conclude that. And we didn't have the data to conclude that. You know, a week after the numbers go up, I hate to say press all the time, but I'm going to say press. The press headlines are, you know, it's a more contagious virus and we're all going to die. And you don't have the data to do that. So I don't think with the exception of Omicron, which is immune evasive, and that explains why it spreads well. None of the others, I'm convinced, are more transmissible or have a higher R naught. Now, the other issue with R naughts is that some people are super spreaders. And there's one paper from studying an outbreak in Hong Kong. And there you can see on the left, patient B infected a lot of other people, more than the 2 to 3 that the R naught tells us for SARS-CoV-2. And these typically happen in, well, you can see family or weddings or temples or bars where there are a lot of people together, and that person is probably shedding a lot of virus, and they're social and infecting a lot of people. And so this, of course, throws the R naught to hell, right? It doesn't fit the R naught at all. So just so you know, super spreading, actually these kind of events accounted for much of the spread early on in the pandemic. Question number 1, which of the following parameters is not influenced by human interventions? Mortality rate, case fatality ratio, reproductive index, incidence, incubation period. Think carefully, which is not influenced by human interventions. Most you got incubation period, which is correct. That's a biological feature of the virus, or I should say a particular virus host combination, because there is a range in an incubation period, obviously, but that's not influenced by what we do for the most part. The others all are. Mortality, case fatality, reproductive index, and incidence. They all are influenced by what we can do. All right, so now let's talk about viruses causing disease. Viral pathogenesis, the process of producing a disease. 2 components: what the virus does to you and your host response. So the virus can kill cells and cause damage, obviously, but in fact, most of the signs and symptoms of virus infections are what the host does in response, the immune response. And we're going to talk about that later on when we talk about immune responses to infection. Viral pathogenesis is a hot area of study in virology. We want to understand how viruses cause disease. So these are some of the questions that we ask. How does a particle get in? What does the host do? Where does the virus reproduce? How does the virus spread? What organs and tissues are infected? How does the host respond later? Interferon antibodies, T cells, etc. Is the infection cleared, where it would be acute infection, or does it become a lifelong persistent infection? And then how is it transmitted to others? And we're going to address some of these today and many of them in the rest of this course. So to be infected, in other words, for a virus to get into you and start replicating and maybe spread to someone else, 3 things. We need enough virus to start with. If you don't have enough virus, it's likely that early innate responses are going to clear the infection. So you need enough virus, and how much is enough depends on the particular virus and the host, of course. We can figure that out in animals in the laboratory, but it's harder to do in people. And this is another topic that was discussed extensively throughout the pandemic. How much virus do you need? And there was one person, who I will not name, who said, when you get more virus, the disease is different. And that's just not true. The disease is not different. It may be accelerated. Things may go faster, but it's not a different disease. All right, enough virus. Cells have to be accessible. The virus can find the cells. They need to be susceptible, which means they have receptors, and they need to be permissive. The virus can multiply in them. And finally, somehow local defenses either have to be absent, and there are some tissues that don't have local defenses, as we'll see, or the virus overcomes them in some way, because most viral infections are actually cleared, or I would say most viruses that come into us are cleared most likely by the innate immune response, which is very effective at stopping them before we know anything. All right, so let's talk about viruses getting in. There are a limited spectrum, and we're going to focus on humans in this course. There's a limited number of ways that viruses can get into us. They're shown on this person here. You have your skin, you have mucous membranes. That's it. Skin and mucous membranes. There are not a lot of ways for viruses to get in. So we're pretty well protected. The skin, biggest organ in your body, right? The outer layer is dead. This is a beautiful defense. The outer layer is dead, so viruses just fall on your skin. They will not reproduce because of that. But unfortunately, so it's a great barrier, but unfortunately many of us have cuts and so forth on our skin. And of course we can have needle sticks or mosquitoes putting viruses below that outer dead layer, which is the stratum corneum. So the whole top part of the skin is the epidermis, and it consists of this outer dead layer, and underneath are living cells. So it's a great barrier, but viruses certainly can get across it. There's a video here I'm linking to that I made, which shows you how mosquitoes spread viruses. So female mosquitoes are looking for blood meal, right? And they probe until they find a capillary. It's really amazing. They're probing and probing, and they find one, and they go in it, and then they start to suck, and the whole capillary collapses as they pull blood out. But as they're probing, they are depositing, if there's virus in their saliva, it's going into the tissues. And they also are putting in anesthetics so you don't swat them and kill them, and other anticoagulants they're putting in. They're a drugstore. Mosquitoes are a drugstore of things that they put in. And then, of course, once they put the proboscis in the vessel, viruses go in there as well. It's really amazing. Anyway, so that's one way viruses can get below the skin. But other ways, you can have a dog bite or a scratch or a needle stick and so forth. All right, so that's the skin. Great barrier, but can be breached. Mucosal surfaces are beautiful because the mucosal surfaces of your respiratory, urogenital, alimentary tracts, they're permeable. They have to be able to exchange things with the external environment. Mucosal surfaces, you got a lot of them. You have to breathe, your cells have to exchange oxygen, CO2, alimentary tract has to absorb. So those cells are exposed. It's a limitation of what they have to do. And so these living cells can be infected. So let's look at the respiratory tract. It is lined with respiratory epithelial cells. The upper and lower tract, they're both lined with these respiratory epithelial cells. They're covered with mucus, which are produced by goblet cells. And they have a basement membrane underneath them, which is a barrier to things getting through the epithelium into the subepithelial tissues where there are lymph capillaries and blood vessels. So it's important to have that barrier. These are ciliated cells, and when we all are inhaling viruses on a regular basis, and they typically get trapped in mucus in the lower tract, and we have an elevator, a mucociliary elevator. These cilia are beating always upwards, and they move the mucus up with the virus particles in them, and then periodically you swallow this mucus, and the viruses get digested. So that's why respiratory viruses often show up in feces. It's not because they're reproducing in your alimentary tract. You are swallowing the mucus. When you have an infection, you're making more mucus, you're swallowing it, and you're digesting the viruses. So viruses typically enter the nasopharynx. They reproduce in the nasopharynx, and many viruses cause symptoms there, and that's all that they cause, upper tract infections. But many viruses can spread down into the lower tract as well and cause a variety of symptoms, of syndromes, including pneumonia. And we know that SARS-CoV-2 can do that as well. Rhinoviruses less frequently, and influenza viruses can do both as well. So that's how the viruses would get in. They would penetrate the mucus layer and begin reproducing in these epithelial cells, which, except for the mucus, of course, are exposed. Then we have our alimentary tract. So by the way, I meant to tell you, we breathe in 6 liters of air a minute. So we're sampling a lot of air, and there are lots of viruses floating around in air, depending on where you are. If you're in a crowded place, lots of viruses. At Penn Station, when people are all crowding to get to a track, you see people putting their masks on because they're still afraid of breathing other people's viruses. So 6 liters of air a minute. The alimentary tract, you're always putting things into it. You're drinking and eating, so you're introducing viruses. And there are also epithelial cells lining the alimentary tract. Here is the lumen of the small intestine. It has a layer of epithelial cells which comprise these villi, and those epithelial cells can be infected by viruses. They also have beneath them tissues that limit the spread of viruses into the underlying cells. Here it is in this high magnification. These are two in the gut. They're called enterocytes, and underneath is a basement membrane, as you can see here. So this small intestine often lets viruses in a variety of ways. They can reproduce in the epithelial cells. Sometimes they are taken up by these M cells. Throughout the epithelium, there are these M cells which are very thin and allow immune cells to sample the antigen content of the lumen. Often dendritic cells go here and put out their processes and sample to see if there are any foreign antigens in the lumen. So it's a very thin cell sheet, and many viruses can get transcytosed through the M cell. Here are some virus particles binding to an M cell. They're taken up through the M cell, and then they're released on the bottom. So many ways viruses can get in through these. Your genital tract can also be a portal of entry. It is protected by mucus, low pH. Sexual activity can cause abrasions that allow viruses to enter in the epithelium. Some of them remain locally, like the papillomaviruses, and some of them can spread from the urogenital tract elsewhere. We'll talk about how viruses can spread in a moment. The eye can be infected. The eye can be infected with viruses acquired from respiratory secretions from other people, from swimming in pools or hot tubs that are not properly sanitized. And there are many different cell types that can be infected in the eye. You can see corneal epithelial cells can be infected with these viruses. The stromal fibroblasts just below the epithelium can be infected. A variety of other cells, even retinal pigment epithelial cells can be infected. And here's would this be a sign or a symptom of infection, do you think? It would be a sign because other people can see it, right? You can see it, but so can other people. That happens when you get some virus infections. You get a subconjunctival bleed. It looks very scary, but it goes away in a few days. I mean, viruses can do that, but other things can do it as well. So if you have that bleeding, it's not necessarily a virus. All right, so the eye is a source of infection. And some viruses can spread from the eye to other places, in particular to the central nervous system. And here we have infections of the fetus that we just discussed in a moment. The fetal and maternal circulations are kept separate, but some viruses can still cross that. Here, by the way, is the syncytiotrophoblast. These are a layer of cells that separate the maternal and fetal circulation. They're fused by syncytin, which is formerly a retroviral envelope glycoprotein, which I told you this in the retrovirus lecture. The gene was taken by mammals many years ago, and it allowed the formation of the placenta. Anyway, there are two terms you have to distinguish, right? Transplacental versus perinatal. Perinatal means around birth. So during birth, maternal blood can contaminate the fetus, and if she has viruses in the blood, it will infect it. That's perinatal. But transplacental is when the virus actually crosses the placenta as the fetus is developing. And the pathogens that do the transplacental passage are called torch pathogens, because this is an acronym for toxoplasma, rubella, cytomegalovirus, HIV, and then other. And recently, Zika virus was added to this list. In 2015, it was discovered to be able to cross the placenta and cause microcephaly in infected fetuses. The outer layer of which of the following is dead but can still serve as a portal of virus entry? Respiratory tract, alimentary, air, eye, skin, or urogenital tract?
What type of defenses do viruses employ in order to get through the stomach acid, functionally, to make it through to the small intestine?
Well, what characteristic of a virus would allow it to resist acids and low pH and high pH bile salts, enzymes through the digestive tract? Well, the viruses that can get through, their proteins are not, right? They're not effective. So most of the viruses that go through the alimentary tract are naked. They don't have membranes, because membranes are susceptible to bile salts. So poliovirus, norovirus, they're all non-envelope, very nicely made icosahedral capsids. But there are some envelope viruses that will pass through. Influenza virus won't. But if you swallow influenza viruses, they'll be inactivated, but the nucleic acid will still be there. So you can still have PCR positivity in your feces, but it's not multiplying. But coronaviruses, many coronaviruses can pass through the intestine, infectious. There are many animal coronaviruses that can do that. I thought this would be 100%. It's the skin. I think that's one person who picked the alimentary tract. So the skin, the alimentary tract, the outer layer is not dead. Gosh, if it were, you'd be in trouble. You wouldn't be able to absorb anything. All right, so we've talked about viruses getting in. How do they spread? Some viruses stay forever where they come in. They remain localized, and they're contained by this epithelial structure. The basement membrane doesn't permit them to spread further. They can move throughout the layer, the epithelial layer. But rhinoviruses, for example, in the upper tract remain there and spread simply from cell to cell. But many viruses go further. They move beyond the primary site. We call this disseminated infections. And if many organs are infected, we call this a systemic infection. And in order for viruses to cross the epithelial barrier, both physical and immune barriers must be breached. And obviously, one of the physical barriers is this basement membrane. And so it turns out that our immune system is to blame for this. When an epithelial cell is infected, immune cells come in to try and clear the infection, and they end up breaking the basement membrane. And so here's an example of a macrophage that wants to come in and perhaps do something with this M cell, and it's broken the basement membrane in order to get in, and virus particles can get out. And inflammation will do the same thing. You don't even need cells here. When cytokines and chemokines are produced as a consequence of infection, we'll talk more about that later, that can break down that basement membrane, allowing viruses to pass through. And of course, if there are antibodies in the mucus on the epithelial side, those could stop infection. So obviously there have to not be antibodies in order to allow viruses to get through. An important component of viral spread is where the infected cell is releasing the virus, the top or the bottom. So in your alimentary tract, or mucosal epithelial cells in general are polarized. They have an apical and a basolateral side, right? The apical is in the gut is oriented towards the lumen. The basolateral is facing the basement membrane. And viruses have different patterns of release. So here, for example, is influenza virus, which is apically released, not basolaterally. And apical release, of course, facilitates transmission in respiratory droplets. That makes perfect sense. Measles virus in the middle there is also apically released. It's a respiratory transmitted virus. But Sicular stomatitis virus is released from the basolateral side of epithelial cells, and that sets up a systemic infection. The virus goes in the blood and ends up elsewhere. It actually ends up in causing a rash, and that can be one means of transmission. And a really nice experimental example of how this works is with a virus called Sendai virus, which is a virus of mice. It's not a human virus, but it's used in the laboratory to study pathogenesis. It's normally released apically. It's a respiratory transmitted virus in mice. But a number of years ago, investigators introduced a single amino acid change in the spike protein, which now allowed the virus to be released both apically and basolaterally. And when you infect mice with that virus, the virus spreads systemically, infects a lot of tissues, and kills the mice. So one amino acid change allows basolateral release, and now the virus has access to other tissues by breaching the basement membrane, as I showed you. And once a virus crosses the epithelium, it can then go to a lot of places. And here's an illustration of that. There's our epithelial surface. If that is infected by a virus, the basement membrane is then compromised by, say, inflammation or immune cells. Bless you. And then the virus comes into what we call the subepithelial space, where there are lymph and blood capillaries. They're very tiny capillaries, and the lymph capillaries are very permeable because their job, of course, is to take up the fluids that are in the areas between cells and bring them through the lymph nodes so they can be checked for foreign antigens. And so virus particles get into the lymph capillaries, they go through the lymph nodes. There they may stimulate an immune response, but they will also get into the blood and they can spread elsewhere. So this is a way that viruses that infect epithelial surfaces can go to many other tissues. And virus in the blood is called viremia. And this is an experiment to illustrate it here. What was done was to inject virus into the tail vein of mice, and then at different days after infection, take some blood out of the mice and ask how much virus is present, say by a plaque assay. So we have virus titer with time after infection. And so you see initially there's a high level of virus there at the beginning. That's what you put in. That's the inoculated virus. So that's time 0. And so when you do an experiment, you always need to have a time 0 and to know that you put virus in and how much you put in. And these days, many people don't do this, and I don't understand why. I always complain about it. And then as you go on, the virus titer drops because what you put in, a lot of it is cleared, and some of it ends up in certain cells, which you're not going to find in the blood. And then you have, at day 2 here, another burst of virus in the blood, which we call primary viremia. This is virus produced by those initial infected cells, and that spreads to other tissues, and they get infected, and then you have a bigger secondary viremia, a typical pattern of infection. Now, virus in the blood is obviously a problem because we have a blood supply made from humans. We can't make artificial blood yet. And so we check blood for the viruses that we know to be dangerous. But no pint of blood is free of viruses because we are full of all kinds of viruses. And if we wanted virus-negative blood, we would not have a blood supply. So we are stuck with leaving nanoviruses and other kinds of viruses in the blood. Yes?
Can you restate what's happening at the valleys here between these titers?
Yeah, so you put virus in the tail vein, and when you immediately sample, that's the virus you put in the blood. And so now you know it's in the blood. You got it in the vein, right? And then that is circulating throughout the animal. A lot of it's cleared. Much of it goes into various tissues where it begins to reproduce. So you have very little virus in the blood. And that first peak is virus released from those initially infected tissues. And that goes back in the blood and spreads to other tissues and infects them. And then you have a second release of virus, which is greater because you've now infected more organs. And here's an example of of the whole course of an infection in an animal model for a virus that is spread by the blood. This is mouse pox. These studies were done many years ago that established some of these principles I've told you. So in mice acquire mouse pox as they're walking on the floor of the forest with their little foot pads. And those have little cracks in their foot pads because it's tough being a mouse. You have to walk all over the place and your foot pads get cut. And there's virus on the forest floor. It's bucolic, the forest floor, right? There's virus from other mice, and so the virus goes into their foot pads. So we in the lab inoculate this virus in the foot pad to mimic as closely as possible a natural infection. So there we have inoculation of the skin. So we're putting a needle in the foot pad, so we're bypassing the outer layer of the skin or the foot pad. The virus gets into the lymph and it establishes a viremia. And then it goes into spleen and liver where it reproduces. And then it makes a secondary viremia. In that graph I just showed you, that would be the second peak. And that secondary viremia brings virus to the skin where you then get a rash. So here at days 8, 9, and 10, you see the rash beginning to develop. And even at this late time, we're just getting the swelling of the foot caused by that initial inoculation many days earlier. And so from putting virus in the foot, it ends up in multiple organs, and you see the outcome in terms of a rash. Humans also get many rashes, of course, as a consequence of virus infection. Here's just a table to give you the idea of what viruses are involved, a whole range of viruses with different names for their diseases. There's measles, there's German measles, chickenpox, shingles, even Zika virus, and of course, they have different kinds of rashes. They have vesicular rashes, like this one, where that kind of looks like a vesicle. That's why we call it a vesicular rash. And then we have maculopapular, which are kind of the flat rashes of other viruses. And here is smallpox, a really bad one that causes a rash across the entire body and can be quite disfiguring. This causes a lot of scarring. This was really a nasty time when we had lots of smallpox throughout the world. But as you know, it's been eradicated and there's no more smallpox disease as of 1979. Next question, which of the following assist in viral dissemination in the infected animal? Viremia, basolateral release from epithelial cells, movement through the lymphatic system, inflammation at the basement membrane, all of the above. That's right. Lecture 12, note the first 100% at lecture 12. All right, yeah, all of the above. Oh, 26, all of you got it. That's good. I was going to say that last person might wreck the percentage, but you got it. All right, so viruses can spread in the blood. They can also spread in nerves, and we call this, of course, neural spread. And nerves have a natural transport mechanism built into them. So here, for example, we have nerves innervating the skin, sensory nerves, which are wired all the way out into the spinal cord, of course. And then we have motor nerves, which are innervating the muscle, and they also go out into the spinal cord. And so viruses can infect these nerve endings in both the muscle and the skin and elsewhere in your body. They can enter nerves, and they can be transported sometimes very long distances. And the transport occurs along the microtubules that go from the nerve ending all the way up to the primary neuron. And here we have we have an example of herpes simplex viruses being transported either in a vesicle or in a capsid, which is probably on its way into the cell body, and this one may be on its way out. And so these vesicles moving by motor proteins on the microtubules move viruses around. And of course, viruses can enter the nerve termini. They can go up to the cell body where they'll reproduce and new viruses will be made. They can be released from the cell body. They can go back down to the nerve terminus. They can be released at a synapse and get into another nerve. So many different ways that they can spread. And that's kind of shown on this photograph here, which is it's a herpes virus which has a green fluorescent protein marker in it. And this is used to actually trace the wiring of nerves in the central nervous system, because you can see that this virus was put in somewhere over here in the lower right, and it's spread synaptically throughout this network of nerves and axons and dendrites. So you can actually map the wiring by using these fluorescent viruses. Now, when we talk about infections of the CNS, we have specific terms. A neurotropic virus can infect neural cells. A neuroinvasive virus can get in from the periphery. And a neurovirulent virus causes disease of nervous tissue. 3 different terms: neurotropic, neuroinvasive, neurovirulent. 3 examples. Herpes simplex. Low neuroinvasiveness, doesn't get in very often, but when it does, it's highly neurovirulent, so it causes disease. Mumps virus gets in often. When we had mumps frequently before vaccines, many kids had mumps virus go in their brains, but no consequence, very low neurovirulence. And rabies is the worst, high neuroinvasiveness, high neurovirulence. And that's one where 100% fatality if you are not vaccinated. All right, so then we want to get into our tissues. We've talked about spread by the blood and by nerves. What about the tissues? So tissues have different barriers to viruses getting into them from the blood. And they're shown here. We have on the extreme on the left where those are endothelial cells where the blood is flowing through. And you can see they have gaps. They're not very tightly joined. And this is very common in liver and spleen and bone marrow. So viruses have an easy time getting from the blood to the liver. In fact, when you inject animals with viruses, one of the first places they go is the liver. Not all viruses will reproduce there, but the liver is very permeable because of this. Next, we have what we find in venules. We have more of a tighter system of joining those endothelial cells together, but they're still pores, but there is a basement membrane around that, and that's typical of those organs that are listed. And then finally, the brain has the tightest organization. We have a endothelial cell with no junctions that viruses can get through, and then there's a basement membrane around it. So very hard for viruses to get into the brain from the blood, although it still happens. It still happens, obviously. And here's a kind of summary of how viruses could cross capillaries from in a number of tissues. We're showing one here where the endothelial cells are tightly joined to each other. So viruses can actually reproduce in endothelial cells, cause inflammation, and they would get across the basement membrane. They can go in lymphocytes. Viruses can reproduce in lymphocytes, and lymphocytes have the ability to get through these tight junctions of endothelial cells. And so they carry the viruses with them, or transcytosis, where the viruses are taken up in vesicles and released on the other side. The brain is very well protected, as I said, from viruses coming out. In addition to the tight junctions in the basement membrane, many of the brain capillaries are surrounded by astrocytes. These are these blue cells here, which serve as another barrier for viruses getting out into the brain. So most of the brain is well protected, but viruses can still get through this in a variety of ways. They can come out of cerebral blood vessels, meningeal blood vessels, and get into the brain proper. They can also be brought in, of course, from peripheral nerves, as we've just talked about. And they can also come through blood vessels in the part of the brain called the ependema, where the cerebrospinal fluid is made. Those vessels are more permeable, and so viruses can get in to the brain from that part. All right, finally, let's talk about transmission of infection from one host to another, and you have to do this to maintain the chain. It's influenced by the virus, of course, how much virus is produced, how stable it is, and so forth, by host behavior, some of which is incorporated into the R0. And a good way to measure transmission is what we call secondary attack rate. And this is the probability that an infection will be transmitted among very specific populations, like a household or in a restaurant where people are confined for a certain period of time, a theater. So you typically do these SAR studies in those kinds of scenarios. And I just want to take another detour here and talk about transmissibility of SARS-CoV-2. If you remember, throughout the epidemic, variants of concern arose, and it was concluded that they were more transmissible, intrinsically more transmissible. But as I've told you, in my opinion, we didn't consider the contribution of human activities to these transmissions, and so you cannot conclude that they are more transmissible. In my view, the spreading of variants of concern, with the exception of Omicron, which is immune evasive. So in an immune population, Omicron will infect you because it bypasses antibodies, and that's why it spreads so well. These variants of concern before that, the Delta, the Beta, the Gamma, the Alpha, they're more fit. That's why they spread in a population in addition to the human behavior. So what do we mean when we talk about viral fitness? Same thing that Darwin talked about. You have a mutation that gives a fitness advantage to birds because of their beak. Same thing applies to viruses. A mutation may occur that makes a virus more fit in a population. It will then outcompete other viruses. So what can change fitness? How much virus is made, particle stability, immune evasion. And during the pandemic, none of these were studied. It was only epidemiological observations that led to the conclusion that the virus was getting more and more transmissible. If you read the Omicron reports, you would think that we're now talking about a virus with an R naught of 50, which just isn't happening. It doesn't make any sense. An example of why it doesn't make sense, influenza virus, antigenic variants arise every year. We're going to talk about this later. The virus changes slightly, it evades antibodies, and those viruses are now immune evasive and they can spread through the population. The influenza people never say they're more transmissible, they're immune evasive. Okay, so when we talk about transmission, there are two general patterns: from an animal to an animal, or from an animal to an animal via a vector. So here we have two humans transmitting. It can be a dog to a human, it can be a dog to a dog, it doesn't really matter. It's just one animal to another. But on the right, we have a vector in between. Here we have a tick spreading viruses among rodents. It could be a mosquito. But the point is there's an intermediary that's spreading the virus. It's not going directly from animal to animal. And when we talk about transmission, we also have terms that we use. We talk about horizontal transmission from one animal to another. So here in this picture, we're transmitting among sheep. All right, that's horizontal transmission. If it's a different species, it's a zoonosis. So if it's from a bat to a human, it's a zoonosis, different species. Vertical is from mother to child. Iatrogenic, something that a healthcare worker does, get you infected. If you get infected in a hospital, it's nosocomial. And then finally, germline transmission is when the virus is part of your germline, like proviral DNA, retroviral integrated DNA is transmitted from parent to child. That is germline transmission. And so there's vertical transmission illustrated on the figure. And to transmit, of course, in most cases, but not all, you have to shed virus. So where can you shed virus? Respiratory droplets, we'll take a look at this in a moment. Skin lesions, those vesicles and rashes can have viruses in them. Blood, if you get cut and someone comes in contact with you and they have a cut, the blood can transmit. Urine, semen, feces can all transmit viruses. So those are examples of where you're shedding the virus and it's finding a new host. And we're going to see respiratory transmission in a minute. You may ask, how do people transmit viruses by feces? Well, we just don't wash our hands. Our hand hygiene is horrible, especially kids who really are good at transmitting viruses. But every time there's a norovirus infection and you get gastroenteritis, it's because you didn't wash your hands and you contaminated your mouth with someone else's feces, or you ate food that's contaminated. All right, so that's all shedding. These examples here are where you don't need to shed to transmit infection. The blood supply, the virus is in the blood, it's put into you. It's not really technically shedding. Intravenous drug use, contaminated needles. Insect vectors, they're putting the virus right in you. Germline is not shedding. And vertical mother to baby, depending on if it's perinatal or transovarial, transplacental. The transplacental, you don't need to have any shedding. The virus is crossing the placenta. So here's respiratory shedding. When you talk, when you cough, when you sneeze, you make lots of droplets. Even talking, I'm making droplets now, and you can't avoid it. especially the plosive, the peas. They make a lot of drops. And you make different size drops. You make big ones that fall to the ground in a couple of feet. You make intermediate sized ones that go 3 to 5 feet. And you make really small ones that can go up to 150 feet. And measles is mostly in these tiny viruses that can go 150 feet, because apparently you don't need a lot of measles virus, and that's all there is in those small droplets. And they can travel a long way. SARS-CoV-2, mainly those 3 to 5 foot droplets. That's why there's a 6 foot distance in the elevators. Stay 6 feet away. It's based on that. It's not exact. It's not an exact science, but it's good. There probably is some transmission through the smaller droplets of SARS-CoV-2, but it's not the majority. The majority is those intermediate-sized droplets. And then nasal secretions on your hands, tissues, subway poles. You can for some viruses, the virus will last long enough so that you touch a subway pole, you pick up some virus, you touch your nose, which we're always doing, you infect yourself. It's apparently not a big deal, not a big way of transmitting SARS-CoV-2. All right, so now, when in an infection can you transmit? These are 3 examples of that. Upper left, the original SARS, 2003. What we're looking at is the incubation period in blue, and then the onset of symptoms. So you can see for SARS-1, you had an early onset of symptoms. Here's virus shedding. So you didn't have the peak of shedding until day 10. By that time, you were really sick, and half of the people were in hospitals, so the infection could be contained. No pre-symptomatic shedding for SARS-1. So no community spread. Nobody walking around infected and shedding, because everyone got some kind of symptom by the time they start shedding. In influenza, there's a little bit of pre-symptomatic shedding here, just a few days or so before the symptom onset. But SARS-CoV-2 is a big problem, because you have an incubation period and you're doing a lot of shedding. You reach the peak of shedding at the symptom onset. So there are many days where you can shed enough virus to transmit and you're not sick. So that's why SARS-CoV-2 is transmitted very effectively. It's transmitted during the incubation period, and also asymptomatically infected people that have never any symptoms. They're moving around and about. They shed virus as well. I'm going to skip this because we're almost out of time, but you might want to take a look at it on your own. 2 more things that control virus infections. One of them is geography. Geography may restrict the presence of a virus because that virus is transmitted by a particular vector, a mosquito, or it has a reservoir host that's only in certain places, all right? So I want to give you an example of this as chikungunya virus, how a vector can affect localization of infection. And here are just 2 maps showing you mosquito ranges in the US, Aedes aegypti, Aedes albopictus. As the climate warms, these ranges go north. So eventually, I don't know how many years it's going to be, these mosquitoes will be throughout the United States, and the diseases that they transmit will be everywhere as well. Chikungunya, a alpha virus, envelope virus spread by Aedes aegypti, it infects you, you get fever and joint pains. All right, causes kind of an arthritis type illness. This virus was always seen in Asia, Africa, never Europe or the US before 2004. Spread by Aedes aegypti. 2004 began to spread to other countries, eventually ended up in Italy, throughout Europe. It's even come to the United States as well. Why is this? So originally the virus is spread by Aedes aegypti, which is not present in Europe or most parts of the US. But a variant arose before 2004, a single amino acid change in the glycoprotein of the virus that now allowed it to reproduce to high levels in Aedes albopictus, which is one of the other mosquitoes that was introduced actually into the US by the used tire trade. Used tires are full of water and they always have mosquito eggs in them, so you bring mosquitoes into your country. This came into the US through Houston and is now throughout many parts of the US. And it can carry this new variant of chikungunya. So a change in the vector changes the ability of this virus to infect certain places. And we've had a number of imported cases of chick in the U.S., very few local. So mainly in Texas and Florida, we have Aedes aegypti that can transmit this virus. But the possibility is that it can enter in Aedes albopictus and spread more extensively. And lastly, virus infections are typically seasonal. They don't occur throughout the whole year. Here we have rubella there in the U.S., which you can see peaks of seasonality. Influenza in temperate climates is a winter disease. And polio was a summer disease. Why is this? Who knows? We really don't understand much of this. There is one set of experiments done with influenza virus that says temperature and humidity influence transmission. And so in this experiment, they looked at transmission among, I think, hamsters in cages. Here they increased the relative humidity. And so at 20 degrees, you have very good transmission. Then as you increase the relative humidity, the transmission goes down, and it goes back up again. But the reason it goes down is we think the droplets the respiratory droplets are absorbing water and they're falling to the ground. And if you do this at 5 degrees, the temperature of the winter, you can tolerate more percent relative humidity, but eventually at high relative humidities, the transmission goes down. So in the winter where we have low relative humidity and low temperature, that's the best conditions for transmission of influenza viruses. And again, the humidity makes the droplets get bigger and fall to the ground sooner, so you don't transmit infection as well. We've got viruses in us. Next time, we're going to talk about the earliest responses to infection, intrinsic and innate defenses.